Yeah, I want to, of course, I'm the one standing in between you and a delicious lunch. It's been really good food here, so I'm sure I have to speed up a little bit for you to actually get there, right? Um, yeah, we're going to talk a little bit more about cell wall synthesis. I did a lot of microscopy bits in the first talk. We're going to talk a little bit more about molecular biology, perhaps, in this talk. Just a reminder, this is basically how the cell wall look like, right? Remember this jungle of mainly sugar-based polymers, right? Coming together around each and every plant cell. I showed you these questions, basically what we're interested in my group. We're going to talk about two other aspects of cell wall biosynthesis or modifications in this talk. I'm not going to focus on development, but rather environment, more specifically salt stress, which also is an important problem in agriculture. I'm also going to talk a little bit about how proteins interact in cellulose biosynthesis, and also hopefully introduce a methodology that might be useful to some of you to understand protein interactions. All right, I showed you this as well. It's a pretty cool movie, I think, in terms of following cellulose synthesis in real time, right? Let's see. And this is basically then how the cellulose synthase complex look like. So you have this central part that contains these cellulose synthase proteins, right? And then you have some additional proteins here that are contributing to cellulose synthesis. And we'll talk a little bit about how we come up with those proteins or the corresponding genes as well. And actually, as a backdrop to this whole story, you can see here that this protein complex actually consists of three different main components. They're called cellulose synthase 1, 3, and 6 in Arabidopsis. Right, so intuitively, these three proteins need to be present in the same cell at the same time to form a complex, right? So if we now take a step back, the genes that encode these proteins would probably be expressed in a similar way as well, right? So if one gene was on, the other two genes would probably also be on. And actually, this goes back again to where I overlapped with Sue at Carnegie. We did these co-expression analysis at the time. So there was actually some of the first co-expression analysis, I think, in plants. But anyway, you can see the expression patterns of these three genes across many different conditions and tissues and organs and so forth, right? And you can see that they're really highly correlated with each other. So that means that these guys are co-expressed with each other. Now, if you now take the next step, if there will be other genes that have the same expression profile, we would assume that they maybe are connected to cellulose synthesis as well, right? And basically what you can do here is, of course, identify those expression profiles. Cool. Now I have plenty of water. And then basically order knockout lines for those in Arabidopsis. That's really easy, right? And just assess whether they're impaired or have defects in cellulose synthesis. Right? Another way, I mean, of course, we can overlay expression profile after expression profile here and look at them, right? But another way of doing this or showing this is through these ginormous networks, right? Or hairballs, if you want, right? So in this network, you would have nodes that represent a gene, and then you would have edges or lines between two genes that then represent a certain co-expressed relationship. And of course, I've zoomed in this ginormous hairball for our SESA genes, and they're really close in this network. You can see them together here, 1, 3, and 6, right? And so throughout the last 15 years or so, we have basically identified many genes that are now really important for cellulose synthesis through this approach. So these are genes that encode proteins that are involved in actually binding to cellulose microfibrils. There are other ones that are basically associating the SESA complex with the cytoskeletal components, and yet other ones that are involved in basically putting together the complex and signaling. So in this talk, I'm just going to nerd out a little bit on the mechanism of one of these. And it's basically following maybe Brandes' talk a little bit, like going a little bit excessive in that direction, I would say. But I think it's important to understand like how we're getting from a protein of complete unknown function to delivering an exact mechanism for how this protein works in the context of cellular synthesis. So to be okay, actually, we need to know as well that the SSAs are connected to microtubules, but I showed that in the first talk already, so that's fine. So in a first study, we briefly outlined a crude mechanism for how this protein works. And basically what you can show here in blue is that this protein, companion of cellulose synthase, maybe not the most original name ever, but it's basically a companion of the cis-A complex. It's hanging out with the cis-A complex, moving with it when cellulose is synthesized. The function of this protein is not becoming present in a sense until we expose plants to salt. And so what's happening during the salt stress is that the cis-A complex is removed from the plasma membrane inside of small vesicles, and the microtubules are falling apart. And this protein is now able to reassemble the microtubules, and therefore this complex can now go back to the plasma membrane and continue to synthesize cellulose. Okay, and this is basically a way for the plants to maintain directed growth during salt stress. And so obviously now in the absence of these blue guys, it's a double mutant of the two main homologs of these, you can now see again the cis-A's are internalized, microtubules falls apart, but this protein that is supposed to like put together the microtubules now it's absent, and therefore no more cellulose is being produced. So this is, of course, like a crude mechanism for how this protein works in the process, right? But we were really interested in the kind of molecular mechanisms behind this. How are the proteins actually doing this, right? And so we focused on this inside part of the protein. So this is basically the part of the protein that is facing the cytosol and interacting with these microtubules. And the reason for that is that by only taking this piece of the protein and putting it back into the double mutant, we can restore growth on this salt-containing medium. So basically what we do here is grow seedlings for 3 days and then transferring the seedlings to salt. And then you can see that the wild type is continuing to grow, but this mutant is not. But if you now just add this piece, you rescue this phenotype, right? So therefore, we were really interested in how does this part of the protein actually work, right? So we did a lot of assays in vitro initially. So we expressed this part of the protein with a his tag. You can see it's produced quite nicely here. And then what we did was to incubate this protein part with microtubules. And basically, I thought this image was really amazing the first time the PhD student showed me, because here is an electron microscopy image, as Mikael has shown you before as well, right? But here is two neighboring microtubules, and our protein that is now labeled with gold conjugated antibodies against this his tag. You can see that it's like in between neighboring microtubules, equal distance, right? So it's basically this protein is like gluing together neighboring microtubules, right? And so just to go back to your cell biology 101, microtubules are small tubes, microtubes, right? And you can see those here, and you can see that they're made of rows of tubulin proteins that come together to form this tube. And so now we wanted to ask, okay, if these guys are gluing together microtubules, where on the microtubule surface are they actually binding? So we did something called a chemical cross-linking experiment. So we basically incubated microtubules with our protein, and then we added a chemical that basically covalently link your protein with the microtubules. And then you can identify these crossovers via mass spec technology. And then we can map down that actually our proteins are binding in this red and orange configurations in between neighboring protofilaments on these microtubules. So now we have like a pretty nice initial mechanism for how the protein engages with the microtubules, right? So we have this CC proteins that aid in this gluing of microtubules and bind neighboring microtubules in between these protofilaments or these rows of tubulins. So how does the protein do that? So for this, we turn to NMR, another method I think you introduced briefly as well, Misha, right? I mean, it's basically NMR. So you label your protein and then you take magnetic pulses that basically like change the directionality or like the movement of your of the amino acids basically in your protein, and each amino acid movement is basically corresponding to a peak in this graph, more or less. And so our prediction of this protein was that it was unstructured. And actually, I'm not an NMR specialist by any means. You probably heard that from the explanation. But anyway, this peak distribution confirmed that the protein is unstructured. So this was nice, but not very helpful. But what you can do now is add microtubules to this. And then you would change the chemical environment of the amino acids that are close to the microtubules. So basically what you now do is like you have your NMR graph without microtubules and with microtubules. And you can see, for example, here, you have a displacement of the peaks. And that means that those amino acids are close to the microtubules. So you can begin to see what part of your protein is actually binding to these microtubules. And now we can nerd out even more, right? And begin to like plot basically the plus minus microtubule signals, and then look at which amino acids are now really in close vicinity to these microtubules, right? And you can basically plot this ratio and then find out that probably you have these 4 microtubule binding domain of a protein. All right, so that gives us basically like this type of image here, right? Where you have the SA complex, you have this like CC protein that now, basically like tethers 2 microtubules together through these potential 4 microtubule binding domains. And actually through more specialized NMR, you can actually nail down which amino acids in those domains are really key to that. And we mutated 2 of those sites and show that indeed those are very, very important for this interaction to happen. Great. Now we have the next step, right? I said we would nerd out on mechanism, and I'm not kidding, right? So the CC1 and 2, they are SESA-associated proteins that maintain microtubule coherence during salt exposure. But how does this relate to salt stress? That's, of course, the next question, right? So key to the next step of this project was basically to find interactors of our CC protein, because we thought that maybe there's additional proteins that are modifying our CC1 in some way to actually like change this during salt stress, or like the interaction during salt stress. And so we did this by simply taking a GFP fusion of our protein and then pulled that down with GFP antibody. And of course, we used a control for this. And then here is just like basically the overlap of positive interactors in response to salt. So we have 0 hours, 2 hours, and 12 hours. And here we were mainly interested in actually the overlap initially between those. So we have 26 proteins to choose from. One of those were Veronia. Maybe it doesn't tell you too much, but in a molecular biology audience, people will be like, whoa, okay. It's basically like auxin. I mean, it's involved in everything. It's one of those receptor-like kinase that if you do anything to a cell, this guy will be associated with it, right? So initially I told the student, like, let's stay away from this because there's so many groups working on this receptor-like kinase, right? But then I talked to a collaborator on another project at the stress center in Shanghai, Chun Chao Chao, and he told me that they also had come to a similar conclusion that Veronia and CC have some relationship. So we continued to work on this project with him. And so the reason why we were also like, okay, Feronia could actually have a function in this, because Feronia, among other things, actually like sensing salts, at least like the cell wall damage that is caused by salt, and then signaling in some way to get a response from the cell to this treatment. Also, you can see if you have Feronia mutants, so where you knocked out this Feronia gene, they're much less tolerant to salt stress than a wild type. So there were some interesting connections, at least. So it's not completely off. So Feronia is a receptor-like kinase. As Brande pointed out, usually you would have like a kinase substrate in that case. So something that gets phosphorylated, potentially our CC1 protein, because it interacts with this Feronia. So the next question was, can CC1 be phosphorylated? Right? That seems to be an obvious next step. Yes, there's actually 12 potential phosphorylation sites. We confirmed many of them ourselves in phosphoproteomic analysis that can be phosphorylated. So that was also a nice thing to show, right? And so then the PhD student and our collaborators went further and confirmed the interaction with the typical like different toolboxes that are available. So we could confirm that the CC1 is interacting with this receptor-like kinase. Okay, so just to remind you, if we're lacking this CC1 protein, we have this internalization of the CSA complex, microtubules falls apart upon salt stress, okay? Remember that. Yeah. Now, if Feronia is important for this process, we would assume that a Feronia mutant would have the same kind of phenotype. And so to test that, we took a microtubule marker, introgressed it into the Feronia mutant, and you can see here when we treat seedlings with salt, the wild type copes relatively well with that, whereas in this double mutant, you can see that the microtubules are gone, and the same scenario for the Feronia mutant. So at least there's a link between how these mutants are responding to salt stress. Right. Of course, the ultimate evidence for the involvement of these phosphorylations by Feronia would be if Feronia can phosphorylate directly our protein. So we did actually a lot of assays to show this. Here's one such assay where you just express the active domain of our kinase together with our substrate, of course, the CC1, and add ATP, incubate that, and you do mass spec analysis to then check that, right? And from those mass spec analysis, we can show that there's at least 6 potential phosphorylation sites that are now added through this Feronia, right? The cool thing to me was that these phosphorylation sites are basically on top of where we previously showed that we have this microtubule binding domains. So this could actually be a neat mechanism for how this protein interacts with the microtubules. Of course, now we generated the obvious constructs. We mutated these 6 sites either to something called phosphonull, so basically amino acids that cannot be phosphorylated, or to amino acids that are mimicking phosphorylation, so to actually have it always on. Right? And then we transform these constructs into this double mutant plants. Interestingly, none of those constructs rescued. So what you can see here is like when we transfer the seedlings to salt, wild type continues to grow, double mutant stalls. The wild type construct, of course, rescues, right? But all of these different phospho constructs do not. And just to, again, remind you of this mutant phenotype, when we now look at the microtubule arrays in these like phosphomutants, you can see that actually both in the null and the mimic, the microtubules are basically gone. So why is this? First, we were like, it doesn't make sense. It could be that the mimic is not really mimicking, but it could also be other reasons. And actually, I promised Brandon to show one Western blot, and here it is. Basically, what we did here was to pull down the CC1 with this YFP. And then we did basically a Western blot with this phosphoserine antibody. So basically to see phosphorylation pattern of our CC1 during the salt stress, right? And what was really surprising too is that actually before salt stress, we have a heavy phosphorylation of this protein. And when we begin to treat this, you can see after 2 hours, we reduce this phosphorylation substantially. And then that phosphorylation is coming back again. And of course, that is lost in the Feronia mutant because there you would not have the kinase anymore. So this actually kind of explains why the phosphomimetic is probably not working, right? Because you need this dynamic phosphorylation pattern to be able for this protein to actually work properly. So just to explain it in a different way, typically under normal conditions, we will have a heavily phosphorylated CC1. And actually this makes a lot of sense because phosphorylation is inducing a negative charge in your protein. And the microtubules are net negatively charged. So they actually repel each other. And if you have a moving protein complex, of course, you don't want to have a tight interaction between your protein and the microtubules, right? It will basically putting a brake on the train on these rails. Yeah. So what is happening under salt is that you lose this phosphorylation, and this is increasing the affinity of our protein to microtubules, and we could show this in the paper as well. And therefore, you now begin to stitch back the microtubules together. And of course, now we're regaining the phosphorylation, as you can see up here as well, when you continue to produce elements. Okay. So this is enough of nerding out on mechanisms, but I hope it's showing you that we can go from a completely unknown protein to probably the best characterized microtubule-associated protein in plant biology through these type of approaches. Right. So we can tell here that Feronia adjusts the phosphorylation of CC1 to control this microtubule behavior during salt stress. Last part of this, I'm going to introduce basically a new toolbox for analyzing protein-protein interactions in plant biology. Maybe this can be of interest to you guys as well. But I would say that protein interactions was central to this whole project, right? Without that, we wouldn't really have these results. And maybe we can also take a step back, and most proteins we're working on, we don't know what the heck they're doing, right? I mean, of course, we can select proteins that are already known and continue to work on those. But most proteins, we have no clue. Even in Arabidopsis, about 1/3 of the proteins, we do not know what the function are of those. And so, of course, if you know what proteins are interacting, and one of those proteins is known, then you can infer a function for the interacting protein. They're likely to be involved in the same process. So it's something called guilt by association. If you're associated with someone, you're guilty by just this association, right? There's many tools to analyze protein interactions, right? There's some that are really, really quite challenging, and you have to invest a lot of energy and money usually as well to do that. And then you have other types. You have, for example, standard techniques that are kind of screens and so forth, yeast-based assays, hybrid assays, right? You have co-IP, pull downs, etc., etc. But we all know that most of those actually comes with a lot of drawbacks. Many of those, I mean, in our hands at least, any split YFP assay, you can get anything you want to interact. And the same thing with yeast. I mean, I'm sorry, but the yeast 2 hybrid, it's great to confirm things, but I would be very hesitant to rely too much upon that. More recent advances have introduced something called proximity labeling. And so proximity labeling is basically outlined here. So you have your favorite protein of interest in yellow, right? And you fuse that to an enzyme through standard molecular biology techniques, right? And so this enzyme can then take a certain substrate here in black and put that substrate onto proteins that are close to your protein of interest. And then, of course, afterwards, everything that is now labeled with these black dots, you can enrich, you can mass spec that and find out what the heck it is. Most methods are actually using biotin bases for this. So this is basically a biotin ligase, if you want. And so this system, that comes with a lot of course, positive aspects. It's an optimized system. You can see all of these different enzymes over here have been developed. So Turbo ID is probably the one that most frequently used currently, right? Because there's many users, right? So the optimization comes automatically with that. But there's some negative or basically drawbacks with this system as well. You typically, first of all, plants have biotin already, so you will have some background anyway. In many cases, that biotin level is not enough, so you have to add additional biotin. If you're working with seedlings, that's probably A-okay, because it's easy to add things. But if you're working with flowers or grain development, then it's relatively difficult to add stuff from the outside that gets into, for example, a grain of an organ. Another drawback is that the substrate is released from the enzyme. So that means that these black dots are released from this enzyme before it's attaching to its substrate. So it will float away from the enzyme. So you would have some false positives in the system for sure. Another system that we are using is fully genetically encoded. Again, you have your protein of interest, called bait here, fused to an enzyme. In this case, it's called path A. In this path A, instead of using biotin, it will use a peptide. It's called PUP. And that PUP is put on lysine residues on proteins that is in close vicinity of your bait. So it's basically a ubiquitin-related system, but this comes from bacteria, and it's not present in eukaryotic species, so you will have no background. Okay, of course, there's positive aspects and negative aspects. The negative aspect is there's less users. Currently, it's basically our group and Yanshin's group in plant biology. So there's certainly room for optimization, right? However, there's some really positive aspects. All of these components can be genetically encoded, right? So you can do this in your grains, for example. You can do it in whatever, right? Because you can just express both your enzyme and substrate. The second part that I think is positive is that the substrate, so this peptide, is maintained with the enzyme until it actually meets the protein, right? So basically these prays. So this protein here, so the enzyme, has to physically interact with the prey to be able to put this peptide onto it, right? So there should be much less false positives in this case. So we have implemented this in a range of different systems now. So basically like in leaf infiltration in tobacco, Arabidopsis self-suspension, and also stable transgenics, both are actually Arabidopsis, rice, and poplar by now. And it works quite nice. So just as a sanity check, of course, I mean, before we're like basically selling this concept, we'll of course want to also make sure that we're getting some sanity behind it, right? So we actually like can identify known components of a protein complex. And so this is a protein complex that we discovered together with Daniel van Dan's group a long time ago by now, but it's called a T-plate complex after one of the proteins called T-plate, as you have here. But it's an 8-member protein complex that is the major driver of internalization of material in plant cells. So we use this as an initial example where we used T-plate as bait, and then we expected to see all of these guys, right, coming down or like being labeled by that. And maybe you can just remember EH1 and 2 and TW42 and 1, right? Because basically what we're now getting in this volcano plot here you have the enriched proteins after labeling. You can see that we're getting T-plate itself, making sense. We have the EH1, EH2, and we have the TW41 and 2, right? And you can see actually that the background here is quite clean. There's actually not much other proteins than the protein complex that we actually anticipated. Of course, the reviewer asked us then to compare this directly against the Turbo ID system because many people are using that. And this graph over here is basically more or less showing you how well enriched protein components towards compared to your bait are. And you can see that actually the Turbo ID is doing not that well because you have actually the lower the value here, the messier that you're getting basically in terms of labeling. But we are performing really well on track with basically APMS, which is basically a double enrichment pull down approach. So this was really satisfying to see. So of course we have done it as well for our CC protein. So coming back to that, there's a red thread through this talk. So we use these different constructs where we fuse the enzyme to our favorite protein, and we have this as a control. And of course we then have our substrate, in this case tagged with a flag that is easy to just pull down for identification of the proteins. And what we can see over here is that we get CC1 enriched, you have the different CSAs enriched, and you have this CSI1, which is cellulose synthase interacting one protein. So this makes a lot of sense in terms of this, right? Of course, there's also other proteins that you can see down here. But if we now look at the way that the CSAs are actually getting to the plasma membrane, it's getting through the ER to the Golgi and then trafficked up to the cell surface, then internalized and potentially recycled. So there would be a lot of proteins that are interacting with this protein complex. It's not as simple as the T-plate complex in this case. Actually, what was satisfying to see is that we actually got the T-plate in this. And you can see that we have T-plate, the WD42, 41, and so forth. And all of the blue ones here are part of this T-plate complex. And actually through, actually this is a quite high-end super resolution microscopy with a spinning disk, we can actually show that this T-plate complex with this additional component surrounds the CSA complex when it's internalized in a plant cell. So we can basically identify here components that are associated with CSA complex when it's becoming internalized into plant cells. And of course, now to nerd out even further on this part, now there's about 10 to 12 people in my lab that works on this puppet assay. So we have a lot of data. And of course, what you now can begin to do is begin to assemble protein-protein interaction networks based upon proximity labeling, right? And so in this hairball again here, right, you would have now proteins and with each of the nodes, and all lines here now basically resemble like potential protein-protein interactions. And of course, the wider that edge is, the more experiments we have found this in. So we are building up now a proximity labeling based protein-protein interaction network. And currently we are implementing AlphaFold based analysis on top of this to actually like try to make sense of protein-protein interaction. I think this is something that is really well needed in plant biology in general to understand how proteins are interacting. Anyway, going to skip that. Yeah, the summary. Yeah, I guess abiotic stress impacts cellulose synthesis, in this case like salt. We also show that Feronia is an important regulator of cellulose synthase complexes during this process in salt stress. We have, actually this is something I skipped in this talk, so we can also skip that. But we have this new proximity labeling tool, Puppet, that I think will be very useful for protein-protein interaction analysis in general in plant biology. So with that, thanking my group, especially, where is it, Shuai, who was like developing the Puppet approach in plants in my lab. And there is the collaborators that we've had for the different projects that I showed in this presentation. So happy to take any questions, and I'm happy to also like end this conference. So thank you very much.
Yeah, thanks so much, Stefan, for the wonderful talk. Now, are there any questions? Maybe I didn't have any questions because it's very vast presentation, but I want to learn. Especially we choose drought stress in sunflower. We make genomic analysis, but proteomic analysis is very uh, huge job. So it is necessary all of us, especially my team. So we want to collaborate you.
Okay,
Second.
we can definitely discuss that. So sounds good.
So Lila.
Thank you. As far as this, I understand this labeling, it is really somehow similar to the high-C genomics, like you label what is nearby, but for high-C method, it's easy to sequence. You just go and you see, and how do you, I just don't know, how do you identify this labeled proteins?
So with the flag tagged substrate that we have, remember, you can use a flag antibody and just pull down everything that is labeled. Then you send it to mass spec, and there you identify the proteins.
So you know all the you know everything. Yeah, you know all the proteins.
I mean, actually, anything that is a sequenced genome, it's like relatively easy to even do proteomics on, right? You just have to map it on the proteome in that case.
Okay.
Many of the experiments are done in tobacco leaves, right? So there we're getting back the tobacco data, of course, right? Then we map that onto the Arabidopsis genome. So it's relatively straightforward, actually.
Okay, so it's like sequencing because I never worked with it. And another question: so when you your one of the first slides, you showed us the transcriptomic data of co-expressed genes, and so I would expect this cellulose synthase would be somehow similar to housekeeping genes. It would be thousands of genes. But then you say, oh, we just took the CC1 because why? Well, how did you identify it among thousands of
That's
genes?
a very good question. Actually, Sue has also developed a lot of these like co-expression tools in the past, and we as well, of course. And housekeeping genes, yeah. I mean, primary metabolism is like a mess in terms of co-expression. I don't believe that you can really find anything useful from that. We were lucky enough, but of course, we targeted perhaps 100 genes or so initially. But it's better to start out with that than 25,000, right? So at least you're like narrowing down the scope. I mean, it's the same thing with the protein interaction. Not all of those proteins will be directly relevant for what you want to do. So you still need to make a selection. And you base that selection upon what seems most likely in context of your process, right? So you will not be like, I'm getting one protein from this protein interaction. That's it, right? You will get like still 100 or so, yeah. But at least you can begin to sort through 100 instead of like the 20,000+, right? That you would have, or in barley or wheat, it will be many, many, many more, right? So, yeah.
Yeah, thanks again, Staffan. I have one question in regard of the cellulose synthase and the interaction with the microtubules at salt stress. Did you check out other stresses where, yes?
Yep. Those
same mechanism?
No, they're actually not responding to those. So we check drought stress, for example. No response. I mean, we use PEG in this case, right? So I guess it's not really like drought stress, yeah. But we also checked heat stress and so forth as well, and there was actually no phenotype for those. So that that comment we got also from the reviewers, of course, they wanted to see whether this was a general kind of response to abiotic stress, but it does not seem like that. In the meantime, we have actually identified 2 other proteins from the same superfamily that also are associated with cellulose synthase complex. They seem to be involved in other abiotic stresses. So it might be that the whole family, it's actually a LIA2 kind of domain on the outside. It's something that is like usually involved in stress responses and protection, right? So,
yes. would be the next question about the Lia protein.
Right. So,
And maybe a naive question at the end from my side about the proximity labeling. You fuse it with biotin, and then it's going on. And do you think that the fusion with biotin has an effect on if you take a possible steric hindering in account? Because you're looking for
you mean in
protein.
terms of functionality?
No, because you would like to check the proteins which are nearby your. But if you would then fuse, your protein of interest with biotin.
I can't hear. Can you leave?
So if you fuse it with biotin, that makes a difference.
Yeah, I mean, the biotin is actually just added on to proteins, right? So you have the fusion that you're making is with the biotin ligase. I mean, in our case, it's of course this peptide related system, right? But, overall, you fuse your protein of interest to an enzyme. And that enzyme is then taking a substrate to put onto other proteins. So, of course, when you put that biotin or our peptide onto that protein, we don't know whether they're functional or not anymore, right? So that we don't know. But for us, it's not really anything we're really interested in, right? We're interested in just identify components.
Yeah. Thank you.
Yeah, thank you for presenting these exciting results. I try to anticipate the perspective of the plant. What is the benefit for the plant to stop cellulose synthase under salt and also to distinguish from other stresses? So to my naive interpretation, I would say the plant just tries to spare glucose for homeostasis for more important things.
Yeah.
And but of course, it's somehow a smart way not to have one mechanism for all stresses, also to distinguish maybe for fine tuning whatsoever. Would you agree to that?
Yes and no. I mean, I guess the why question is usually a very important, difficult question to address because we don't understand the plants that well, right? But I think also like the conditions that we're using in a lab is completely different than what you would have in the environment, right? So these proteins, we assign them certain function based upon observations that we do under ideal conditions, right? In the natural condition, it might be quite different in terms of how these proteins are actually acting. I think the mechanistic details of how it's acting, in terms of binding microtubules and so forth, I think that is probably more robust in that sense.
Without interfering with salt tolerance all too much, because I assume it's a quantitative mechanism. It's not kind of digital thing.
I think, to be honest, I think this mechanism will probably not be very fruitful to to implement in a crop plant. I don't think so. We try to overexpress the protein to see if it's becoming more efficient. Not really. So I don't think that this would be beneficial in a sense. It's pure fundamental research to try to understand the biology. I think in terms of actually applied aspect, I don't think this would be that useful. So. Anything else?
Yes. Staffan Persson, thank you very much, first of all, for your very interesting report. I'm plant breeder working here at the Department of Plant Breeding and Seed Production. We usually select plants on their phenotype traits in the field, but your study is the same, maybe the same, but at the level of molecule. Could you watch there? I saw there were some seedlings under different doses of salt. They were different on their genes.
Mm.
Different way as they develop under salt. Would you watch the was there any relation with the weight, with the index level, influence on the economic trades in the future the plans? Would you watch? Think.
Sure, thank you. As I said, in response to Jochen's question as well, I think, yes, we could probably do that, but I don't think that the mechanism I described here would be that relevant to salt trait in natural conditions. We're usually doing this on seedling or media plates, and I think it's very different than what you would have in the field. So we're trying to understand how biology works. That's our purpose with our studies. Then, of course, like some of the tools that we're generating, we hope that people can take advantage of in the applied field, right? But our goal is basically to understand how plant biology works. So I think in terms of there's still like a bridge between the fundamental research and the applied research, and of course we are working to try to bridge that to some degree, right? But we still have a long way to go.
Firstly,
Mm.
thank you for your speech. And in your speech, you talked about interconnections of proteins, as long as I understand and understood. And you learned interconnections under salt stress, but you've also talked about that you also learned under other stress like heat tolerance. And I'm learning and I'm working in the sphere of plant breeding, of which are I mean, which should be tolerant under high temperature. That the question is that is there any perspectives that learning protein interconnections or protein collaborations between each other? I mean, of heat tolerant under heat tolerant.
Sure.
So you get.
Yeah, sure. So we are doing some experiments as well where we take proteins that are associated with stresses, and then we fuse it to this enzyme that I talked about. And then you can treat your plants with or without stress and see how the protein interactions change. Yeah? So that will teach you a little bit about like how the important protein in that process is changing its function as well. So that's something that we are working on as well. We've been studying, for example, some kinases that are involved in sugar signaling, and then you can also get basically how are the interactions of that protein changes when it has sugar or no sugar. And actually, since that is a kinase, you can also look at the phosphoproteome. So basically the phosphorylation of the proteins that it interacts with through this system. And so that gives you a very nice way of getting potential signaling mechanisms for the protein. So hopefully that makes sense.
Then the first ever step would be learning phosphorylation. Yes.
I mean, if you work on a protein that phosphorylate other proteins, then that would be important because that's usually how signals are progressing. Right?
Okay, thank you.
Sure.
I think, as I understand correctly from your question, you also want help from him if he could guide or like kind of collaborate to do something regarding your tolerance experiment or what? O'zimni dissertatsiyam bo'yicha shu o'zimga taalluqli joyini so'rab oldim-da. Kelajakda kerak emas, rahmat. Yetarli. Okay, then I think, yeah. Thanks so much, Stefan, for
Thank
the
you.
wonderful discussion and yeah, insightful results. And yeah, we can give big hand. Yeah, it's unbelievable. We come to the close of the workshop. Yeah, and now how to say, what to say. Actually, a lot to say. Yeah, to take this opportunity, I would like to thank each of you for your contribution to make this happen. And first of all the invited speakers and for sharing their excellent expertise by giving presentations. It's really, yeah, really exciting. And also for the participants and also I think your wonderful presentations and exciting discussions make this workshop really valuable and engaging, really. Yeah, and also I want to thank to supporting institutions and Pushtaka and abroad. Please come here because without this support, it was difficult. Actually, the idea come up last year that we want to do like workshop, invite well-known scientists and kind of make it collaboration or make it a connection. Okay, then I think we can easily do it, but it turned out not easy. So I thought like I expected if we share on this idea, then everyone kind of excited and come to support us and okay, let's do this. But it was not as I expected, but anyway, we made it. So I'm really kind of thankful from everyone who helped directly, indirect way. Yeah, Shteke, nima aytmoqchimiz?
Ko'p narsa demoqchiman. Okay, first of all, I'd like to thank to lecturers who came from different parts of the world. And we know that the knowledge on biotechnology nowadays is very crucial to improve the crop productivity, tolerance, resistance. So this is important for better crop yield and a better life. Because we have in Central Asia, especially big problems now, including the problem of the water deficiency, heat tolerance, and the other problems. So by the help of the modern biotechnology, we can solve very fastly and aimly we can fight against this kind of problems. So I will continue some opinions in Uzbek. Nima demoqchiman? Avvalo, birinchi navbatda mana shu seminarga kelgan barcha haligi tinglovchilarga minnatdorchiligimizni bildirmoqchimiz, rahmat deymiz va alohida rahmatimiz, tashakkurimiz demak ma'ruzachilarimizni barchasi mana shu molekulyar genetika, molekulyar biologiya, biotexnologiyani dunyoda eng ilg'or markazlarini eng yetakchi mutaxassislari bu olimlar bilan biz hadeganda haligi aloqaga kirishib ular fikrini olish imkoniyatimiz yo'q, bo'lmaydi ham. Bu juda ham katta imkoniyat. Avvalo mana shu seminarni tashkil qilishda asosan mablag'lar bilan yordam bergan hukumatimiz har xil fondlaridan yordam oldik. Rahmat deyishimiz kerak, tashakkur deyishimiz kerak, sababiki bizga hozir biz o'zimiz bilimimizni kuchaytira olmaymiz. Albatta, O'zbekistonda ilmiy tadqiqot kuchayapti. Bu juda ham quvonarli holat. Zamonaviy bilimlar bizda ham mana ilg'orligi kuchayishni boshladi, lekin biz hozir dunyo darajasi bo'lishimiz uchun albatta mana bugungi kelgan ma'ruzachilar yordami kerak. Ular bilan biz birgalikda ishlasak, hamkorlikda ishlasak, yoshlarimizni jo'natsak u yerga, ular o'rganib kelsa, ular bizning kelajagimiz. Shu o'zimizdagi kelajakdagi hamma muammolarimizni hal qilishda shu yoshlarimizni ilmiy salohiyati bizga yordam beradi kelajakda. O'ylaymizki, bugungi o'xshagan seminarlar kelajakda ko'proq bo'ladi. Endi hozir albatta bizda haligi yozgi ta'til vaqti bo'lgani uchun biz vuzlar bilan haligi ko'proq yoshlarni jalb qila olmadik, lekin kelajakda endi hozir niyat qilyapmizki, buni bunaqa seminarlarni seriyali qilsak va bundan tashqari o'sha ko'proq talabalarni ham jalb qilsak, bu albatta o'zimizga, yurtimizga foyda. Hammalaringizga yana bir bora rahmat deymiz. Abrorga ham katta rahmat. Katta yordamchi bo'ldi. Bir-birimizga katta yordamchi bo'ldik.
Birgalikda. Assalomu alaykum, hurmatli mehmonlar. Sizlarni ko'rib turganimdan xursandman. I want to say thanks for our guests. I think our workshop is very informative and very successful. Barnose, yes, organize this workshop. It was very difficult, but we did this workshop. It is, I think, very necessary for us. All our presentation, it is very informative and very useful for us. Our republic is one of the important sector is agrar sector, because our population about 60, maybe more 60% our population work in agrar sector. Because aim of our research is developing this agrar sector. This biotechnology is, I think, it is future in this sector. I want to say thank you very much. Aim of this workshop is learn new technologies, take new informations, and I want in future collaborate with you. Not only me, all our institute wants collaborate with you. Thank you very much. Tashrif buyuruvchilarga kattakon rahmat. Kelib shu ishtirok etganlaring uchun. Qushda ham ko'p narsa aytishdi. Bizim maqsadimiz faqat shu bitta institut emas, butun institutlar kelib, o'rganib, nima deydi, mana shu O'zbekistonni agrar sektoriga, mana ilm faniga rivojlanish, mana ko'rib turganlaringdek, mana takvachilarni juda yuqori darajada biz bular bilan hamkorlik qilib, faqatgina hamkorlik orqali shu darajaga yetishimiz mumkin. Agar biz o'zimiz harakat qiladigan bo'lsak, uzoq vaqt talab qiladi. Biz bu darajaga yetkazimizcha bu olimlar yana ham yuqori darajaga yetib ketadi. Eng yaxshi yo'li borib o'rganish, tajriba, workshoplar tashkil qilish, hamkorliklar loyihalar qilish juda muhim hisoblanadi. Shuning uchun shu vaqtlaringni ajratib kelib, o'rganib, mana savol javoblardan ko'rinib turib, qiziqib qatnashganlaring uchun rahmat katta.
I wonder if anyone from the speakers or from the participant want to share their opinion and or impression about the workshop. If someone want to say something from the speakers. Speak.
Thank you, Barno and Abror and everybody here. It's been really wonderful to come to Uzbekistan and Tashkent and meet many of you. And I'm really excited to continue the communications and potentially collaborations. And I've met a few students here who are amazing. Arno is amazing. I feel very privileged to cross paths with Barno, and I'm looking forward to seeing her continue her success in her career, and she will. I have no doubt that she will become one of the leaders in plant science in the world, actually. And so I'd love to meet more Barnos in Uzbekistan, and this is my first time here, and my impression is really fantastic. And thank you so much for the opportunity and the privilege to come. It's been an honor. And I hope, like I said, that this is not the last time I will be engaging with you guys.
Thank you. Wonderful.
So for me, it's very easy. I fully agree what you have said.
Sure. I've also enjoyed the workshop, and I hope there will be more workshops in the future. And I hope that in future workshops, there will also be presentations from the local researchers here working in the two institutes and the university so that we can communicate further and interact more with all the great research that's going on here. So thank you very much. I think this was a first step, and I'd like to see more activity in the future. Thank you.
Yeah, of course, I agree. There's not much more to add, I guess, what already has been said. But I also hope, in context of microscopy and cell biology, I hope this is also, maybe a good starting point to develop something advanced here as well. And I hope that will happen. I also have to say that I've been eating too much good food. It's making an impact, I think. So thanks. No apologies needed.
Yeah, I fully share what the other speakers have already said. My observation was that there are particular persons here in this room from Uzbekistan who are extremely interested. And when I have a maybe more broad perspective, it's obvious that your country needs biotechnology, plant biotechnology, as a breeding tool, because you had a lot of drought, you have too less water, and you have to cope with that. And this problem exists all across the world, but here, of course, it's a particular problem. And I see that we could contribute that via cooperation, via information flow, via communication, we can just make a better world and a better country.
Я, если вы позволите, говорю по-русски. Большое спасибо, Барну, за этот за замечательный воркшоп. Большое спасибо всем участникам. Спасибо, Курши, спасибо, Аброр. Спасибо всем, кто пришел. И я хочу, конечно, хочется сказать, мне нечего добавить к словам моих коллег, но на самом деле я бы сказала, что не только с нашей стороны мы можем внести какой-то вклад в дальнейшее развитие Узбекистана, но я абсолютно уверена, что Узбекистан, поскольку является действительно засушливым регионом, он несет в себе очень ценные генетические ресурсы для того, чтобы в дальнейшем обеспечить и помочь всему миру бороться с засухой. So I wanted to add to Johan's speech that Uzbekistan, as the same as Brazil, being hot region, definitely has lots of genotypes and genetic resources, which also could contribute for European development of this drought and heat resistance research, and I believe for future cooperation will be very productive. Thank you.
Я тоже скажу на русском. Я тоже поблагодарю Барна, Куршида, Аврора. Вы вообще сделали очень замечательную работу. В общем, пригласили столько крутых ученых со всего мира, и не на каждой конференции вообще можно услышать по целых 2 лекции, и таких полезных, и вообще современных. Это вот пик науки, пик биотехнологии, генетики. Это вообще очень ценно и очень здорово.
bitta narsa aytib qo'ymoqchiman, uzr esimdan chiqib gapiraman deb. Demak, Barnoxonga ham bitta chapak chalib qo'ysak nima uchun? Bu olimlarni biz o'zimiz olib kela olmaslik, ochig'i. Demak, Barnoxonning yordami katta bo'ldi mana shu masalada.
Hello, everyone, again. Let me speak firstly in my mother tongue. Assalomu alaykum yana bir marta hammaga. Demak, bugun mana afsuski workshopimiz, mana 3 kunga bag'ishlangan workshopimiz tugab boryapti. Bu workshopda mana genom tahrirlashdan boshlab tortib u demak molekulyar o'simliklar molekulyar seleksiyasigacha, strategiyasigacha bo'lgan nimani o'rgandik. Juda ko'p narsalarni, yangiliklar bilan tanishdik. Endi bu workshop mana hozir yuqorida biroz eslab o'tildi. Anaqa oson bo'lmadi bu narsani qilish. bular uchalovlarini anaqa asab buzilishlari, bu katta narsa. O'rtaga men ham tushib qoldim orada. Bular o'zimiz bilamiz u narsalarni. Lekin nima bo'lganda ham men Barnoga, Xurshid To'raqulovga, Abror Qurbonovga o'z minnatdorchiligimni bildiraman. Bu workshopni tashkil qilganligi uchun. Bu workshopni biz 1 yil oldinlar taxminan demak rejalashtirgandik. Mana bo'ldi. Bu dunyodan ko'plab nimalarni, olimlarni, yetakchilarni olib kelish o'zi bo'lmaydi. Demak, thank you very much for our speakers from diverse countries, as well as from Denmark, from Germany, from United States, and from Russia, and from our brother country, Kazakhstan, and I would like to thank in your mother language. So Gamsamnida, Dr. Sang Yanri, yes, and Mangtak, Brenda Wolf, and Stefan, and Danke schön, Dr. Johann Kumnen, and Michelle, and спасибо огромное, Sofia and Nastya, and Rahmet Kanatava, and very good, very excellent presentations, and very exciting discussions, not only here, I mean, not only presentations during presentations, and also in coffee breaks times, and lunch times, and I'm very exciting. Thank you very much.
Rahmat sizga. Kanat, you really sit with next to like a speaker. So I again you forgot and you didn't see it. So I'm 2 times. I'm really big. I'm really sorry, Kanat.
That's okay. So I'm not. Thank you very much. Yeah, it's has been a fantastic 3 days. I'm happy to meet my previous colleagues and have a chat with them about the great science they're doing. I'm very happy, and just don't worry. I feel myself, as a local, so I don't get upset, and no worries, please. So what I can say is, whenever and wherever I meet any Uzbek brothers and sisters, I get quite amazed by their enthusiasm. So this is what we want to learn from you guys. Thank you very much for the exciting workshop.
Öncelikle tüm Özbekistanlı kardeşlerimize, Kazak kardeşlerimize ve dışarıdan gelen misafirlerimize diyeyim. Hepsine çok teşekkür ederim. Buraya davet edildiğim için ve organizasyon komitesine, özellikle Ebrar Bey'e çok teşekkür ediyorum, Pamuk Araştırma Enstitüsü'ne. I want to be excited to be in here. They invited me as a speaker. It is a very big experience for me because I see many experienced scientists in here. So it is a very high level. I want this kind of workshop will be in the future in Turkey, the other countries, and I very excited the next step. Thank you, everybody. What I can say at the end, I hope that the connection we made here
will grow into fruitful partnership in the future. And thank you, all of you, for joining and making this workshop wonderful. Can we make a last photo? Yeah, group photo, group photo, group photo. Then after this we have lunch break. Yeah. The lunch break is the same place, right? Good.
That's good.